Today we are going to start the last part of this lesson, of the lesson of molecular genetics. And we are going to see 4 different techniques of biotechnology. First of all, it's going to be cloning, electrophoresis, PCR, and genetic manipulation. We are going to see how we can clone individuals or clone some cells for therapeutical treatments. Also, we are going to see what is electrophoresis, which is a technique that allows us to recognize certain DNA fragments. Then we are going to see what is PCR, the famous PCR, okay, which is a technique that allows us to reproduce or duplicate thousands of times the DNA. And lastly, we are going to see genetic manipulation. So let's start with cloning. I'm going to explain you first of all the cloning of individuals. Here we have an example with a sheep, because the first mammal that was cloned was a sheep. The cloning little sheep was called Dolly. Okay, it was the first clone, the first mammal clone. And let's see how it's made. Joder, perro maldito. Okay. For cloning, we need, in the case of a sheep, okay, we need the sheep that is the one that we are going to clone. Okay, we are going to obtain a sheep that is going to be identical, genetically identical to this one. Then we need another sheep, which is going to be a ovum donor. Okay, it's going to donate a ovum. And finally, we have the third sheep, which is the one that is going to subrogate the maternity. Subrogate the maternity is that like the vientre de alquiler, maternidad subrogada. Okay, so it's going to we are going to use her uterus for develop an embryo. Okay, so first step, we need to take a stem cell, una célula madre, from the sheep that we want to clone. What is a stem cell? A stem cell is a cell that is undifferentiated. Okay, it has at the beginning it has no difference. It's not a cell from the skin, it's not a cell from the eyes, it's not a cell from the liver. Okay, it's undifferentiated cell. And the stem cells can create any kind of cell, okay, of our body. The most famous stem cell is the zygote, okay? The zygote is a stem cell. You know that when an ovum and when a spermatozoon fertilizes an ovum, it creates a cell which is called zygote. It is a stem cell. Then that cell is going to divide into more stem cells, and those stem cells are going to differentiate for creating all the individual, in this case a sheep. So we take out the stem cell. The stem cell, here I have written 2N. What does it mean? It's diploid, okay? Diploid, not like the ovum. The ovum that we take out from this sheep, it is N, not 2N, because it's haploid. You remember that. Gametes are haploid. They need to join together to create a diploid organism. Okay. So for creating an organism, we cannot use an ovum because it is haploid, but we can use a cell, okay, an adult cell, adult stem cell in this case, which is diploid. The first step, now we have to take out the nucleus of the ovum. this ovum has a nucleus, and we are going to take it out. And then we have the cell, the ovum, with no nucleus. And then we take out this nucleus and we put it in the ovum. And now we have an ovum, a gamete, which is diploid, nor haploid. This ovum is going to divide and is going to create an embryo. We put it is going to start dividing and creating an embryo. This is what we call in vitro, because we do it in a crystal, okay? In a crystal recipient. And finally, this embryo we have to put in the uterus of the suffragant sheep. Okay, this is the suffragant sheep, or the, what do you say, the vientre de alquiler. Okay, we put the embryo into this sheep, and then after some months, it's going to be born a little sheep, and this little sheep is just the same as this one. It has the same genetic material. It has the same genes. We say that this sheep is a clone of this one. This sheep is genetically identical to this one. So this is cloning for reproducing individuals. Obviously, this is forbidden in humans, okay? And this have been done in sheep, in monkeys, in a lot of animals. But in humans, what we can do is the therapeutical cloning. What's that? Imagine this is a person and it has burned his skin or her skin, and it needs a skin for new skin, okay, a transplant from another person. But you know that when you receive an organ from another person, it's going to be a reject. Our body is going to reject it because it's not from us, so the defenses are going to attack it. So if we use our own stem cells, we can create tissues for us. Imagine, I burned my skin, so we can take a stem cell from another part of our body, okay, from my body, and then they can reproduce the stem cells, and those stem cells can create any kind of tissue. So they can create skin, and those skin can be implanted into my damaged skin. The same can happen with some organs. In the case of skin, is easy. In the case of an organ, it's more difficult, much more difficult. Okay, so that's the therapeutical cloning. Okay, now we are going to see what is electrophoresis. I told you that electrophoresis is a technique that allows us to recognize DNA. Electrophoresis is done in a gel, okay? And how does it work? It works, it has a current, an electrical current, okay, that is going to move the fragments of DNA, okay? Imagine there's a murder, okay? And we in the victim we can find that there are some scratches. So maybe the killer, okay, the killer has left his some of his cells in the scratches of the victim. So I take the I take a sample from the scratches of the victim, and there I have the DNA of the killer, okay, or the murderer. Here I have the DNA here, okay. So what can I do with this? Not a lot, but I know that the victim didn't get on very well with another person, and I think that maybe this person is guilty for the crime. Okay, he's maybe the criminal. So here I have the DNA that I found in the victim, and there is the person the person whom I think is the criminal. So here I have his DNA. Okay, here I have the DNA that was found in the victim, and here I have the DNA that belongs to the person that I believe that is the criminal. So I'm going to put the DNA of the victim here and the DNA of the person which which uh, which is probably the criminal here. Okay. Okay. Now there's going to be different fragments of DNA because we use restriction enzymes. Restriction enzymes, by the definition, are enzymes that cut the DNA in certain sequences. Okay, they cut the DNA in certain sequences, and those sequences among people are different. Okay, are longer or shorter. So this electrical current is going to move the DNA fragments. And they are going to appear like this. And in this case, the same. The fragments, different fragments, are going to be moved by the current. Why do they stay in different places? Because the shortest fragments are going to move better, and they are going to reach the top, and the heavy fragments or the longest fragments are going to stay here because they have more weight and it's more difficult to move them. Okay, so in this case the shorter fragments are going to move to the top, and this is the result. Now I can say that this person is not the murderer, is not the killer. Okay, because the fragments of DNA of this person are not the same as this one, as you can see. If it was the killer, we should see the same fragments in the same places here. Okay, so electrophoresis is for recognizing DNA. For what purpose? For determine if a person is a criminal. Okay. Also, we can use it in paternity tests. Okay, the paternity test for knowing if someone is the son or the daughter of the father. Okay, And also we can use it for recognizing the DNA of a microorganisms, for example, viruses. And this is what we do for coronavirus. When you suspect that someone suffers from coronavirus, you take a sample from the nose, okay, and then you put it here. We know that the sequence in the case of coronavirus is going to be like this, okay, like this. And then the sample that I take from the person that I suspect that that suffers from coronavirus, I'm going to put it here. I put it here. I do the electrophoresis, and obviously this person suffers from coronavirus. Okay, so electrophoresis is for identifying microorganisms, identifying paternity tests, and identifying criminals in criminals. Okay, and now you are going to ask me, what about PCR, the famous PCR? Okay, the famous. Okay, no. So I told you that I had here DNA from the virus or from the person. Okay, but do you think that if I take a little stick and I take a sample from the nose, I have enough DNA here for doing this? Do you think that if I take a sample from the victim of a murder, do you think that in that sample I have enough DNA here for doing this? No, it is not enough. It is a really small quantity. So we need to duplicate thousands of times that DNA in order to have enough quantity. That's the PCR. PCR comes from the polymerase chain reaction. And you should remember that the polymerase, okay, polymerase is the enzyme that duplicates the DNA. So what is the PCR? In a PCR we need a machine which is called thermocycler, okay? Thermo of that comes from the temperature, okay? And cycler of cycles. I take the sample of someone's nose and I have here the DNA. In the thermocycler I have to put the primer, which is a sequence of DNA of the virus, which I know. I put it here, the primers, okay? I put the nucleotides in order, because I have to duplicate this, and this process is going to be just like DNA duplication. And I have to add the DNA polymerase, okay? The polymerase. I add nucleotides, primers, and the polymerase. Now what I have to do is to rise up the temperature. When I rise up the temperature, you know that the temperature you have learned in chemistry that when you rise the temperature, the molecules are going to separate. You know that. And when you turn the temperature lower, the molecules are going to join together. So if I raise the temperature, these molecules of the two strands of the DNA are going to separate. And now the primers are going to join to the correspondent sequence, because they are complementary. We know that they are complementary to certain sequences. The primer is here, the polymerase comes here and here. And what is going to happen? That the polymerase is going to take the nucleotides and is going to add it. And it's going to create two new strands. The DNA has been duplicated. Then I have to, these strands are going to be separated, okay? And now I have to download the temperature, and when I download the temperature, the strands join together. And at the first step, I have one strand. And now I have two strands. This is one cycle. At the following cycle, I raise the temperature. These two strands are here. I have one strand, no, two strands, one DNA molecule. Here I have four strands, two DNA molecules, this one and this one. At the following cycle, I raise up the temperature. These two strands are going to separate, and these two strands are going to separate, and it's going to happen just the same. Polymerase is coming here, the primers are going to stay there, and they are going to be duplicated again. This one from this one, and from this one is going to appear this other one. The same with this one and this one. At the third cycle, I'm going to have 4 DNA molecules. And now it's going to grow to be duplicated in an exponential way. At the following cycle, it's going to appear 6. No, not 6. It's going to be exponential. So 8. At the following, 16. And then 32. So in a certain number of cycles, I'm going to obtain thousands of copies of DNA. And with those copies of DNA, I can work. I can do this, okay? So the PCR is a technique which allows us to produce thousands of copies of DNA. And when we have those thousands of copies of DNA, we can work with it, okay? With it. That's the PCR. But the PCR doesn't identify the DNA. It's the electrophoresis, the technique that allows us to identify the DNA. Okay. And finally, we are going to see the genetic manipulation. What is genetic manipulation? To take a certain gene, which I'm interested in, and put it in another organism. The best example. You know that people and mammals in general, we produce insulin, and that insulin is a hormone that downloads the sugar levels in blood. And some people which suffer from diabetes, they can't produce insulin and they have to inject it. How do they get that insulin? How do we produce that insulin? And maybe you are thinking they produce it in a laboratory. Yes, but how? Really easy. We take the cells of the pancreas, okay? You know that the pancreas is the organ that produces insulin. So we take the cells from the pancreas. Those cells have DNA in their nucleus and they have the gene for the insulin. This gene, I have to cut it exactly. I have to cut exactly the gene. Yep. How can I cut it? With restriction enzymes, okay? These scissors are not scissors, as are restriction enzymes. So we cut the gene of the insulin. And now I have to work with that gene, so I have to make several copies of it. So I have to do a PCR. And now, this is the gene. I do the PCR and I get thousands of copies of this gene. Okay? Now, we take the gene and by PCR have this. These several copies I have to put inside in another cell. So how can I do it? In the case of a bacteria, we can use a vector, okay, which is a virus. A vector is something that transmits something. For example, mosquitoes are vectors of malaria. You know that malaria is an illness, and the one that transmit that is an illness that is caused by a protozoon, and that protozoon is called I forgot the name, and it's something falciparum. No, falciparum is the name. Yes, so that's the protozoon, and the ones that takes the protozoons from the water and take it and put it inside our blood is the mosquito. So the mosquito is the vector. The vector is the one that carries it. So I have to put this DNA inside viruses. Viruses are going to be the vector. This is not really difficult to do, okay? Because viruses are really simple. And the vector is going to infect, you know that viruses infect cells, okay? They put the genetic material inside the cell, and the cell is going to read this genetic material and reproduce the virus. But in this case, what is going to happen? This genetic material is not the genetic material of the virus, it's the insulin gene. So, this is a bacteria, okay? A bacteria with its own DNA. And here I have the insulin gene. I'm going to put it in a different color. Okay. The insulin gene in black. The virus is going to infect the bacteria and put inside the insulin gene. And this gene is going to go into the nucleus. The nucleus, no, sorry. Bacteria haven't got nucleus. They have DNA, but not nucleus. It's not surrounded by the nucleus. Here we have the insulin gene. And what's going to happen now? What happens everywhere, in every single cell? This gene can be copied into RNA, into messenger RNA, and this messenger RNA is going to be read by the ribosomes. And when the ribosomes read the RNA messenger, they create a protein. In this case, which one is the protein? Insulin. And now maybe you are asking, okay, you have taken the gene, you have reproduced it by a PCR, you have put it into a virus, and the virus infected the cell takes the DNA into its own DNA, and now this bacteria is copying the DNA into RNA, transcripting. Remember, this is the transcription, and then the traduction. During traduction is going to make insulin, okay? And maybe you are thinking, but it's only one bacteria, so it's a little quantity. Don't worry. Bacteria divide every single 20 minutes. So this bacteria is going to duplicate its DNA and then divide, creating two bacteria. And those two bacteria then are going to divide, and now we can cultivate, okay, we can um, farm, okay, we can farm, podemos, no sé cómo decirlo en español, de granja, podemos criar, vamos, podemos criar bacterias. We can, so we are going to have a lot of bacteria, an enormous tank full of bacteria that are producing insulin. Okay, this is how we modify genetic how we modify organisms for producing substances. And now you are thinking, okay, can you modify a person? No, an adult is impossible, because here I have modified a cell. I have taken a gene and put it inside a cell. One cell. I can modify one cell, not a person. We have thousands of cells. Thousands, not millions of cells. It's impossible. But we can create genetically modified organisms. For example, you know that there are genetically modified plants that we use in crops, and also animals. In this case, we do it with the embryo, okay, with the zygote. In the case of an animal, okay, the mother and the father, yeah, because women can have trousers and short hair, and men can have long hair. And also a skirt. Why not? Okay? We have the mother and the father. The mother produces ovumes and the father spermatozoons. Okay? We take it out, and in vitro we fertilize this and we get a zygote. This zygote with a nucleus and its DNA can be modified, genetically modified. In the case of humans, this is forbidden, obviously, because if we could do it, people with a lot of money could have the sons they want. Okay, it's like, no, I want my son to have to be stronger or to be taller. So this is not ethical in the case of humans. Okay, but in the case of animals and in the case of plants, in the case of plants, this would be with a seed. Okay, a seed is a zygote, so we can modify it. We can modify the zygote of an animal and the seed of a plant. So the purpose is that we can create animals and plants that are genetically modified, and if they are genetically modified, they are going to be stronger. For example, in the case of plants, they can be resistant to plagues or resistant to some illnesses, or even they can grow with less water. And in the case of animals, there are a salmon which is genetically modified for growing faster. They use this salmon in piscifactorías. I don't know how to say that in English. Okay, no, fish farms, I think. Okay, they use this salmon in fish farms. Okay, and these salmons grow faster because they have been genetically modified. They inserted a gene that is for growing faster. And maybe you are thinking, okay, that's all right. Genetically modified organisms are not harmful, okay, okay? If you eat corn that has been genetically modified, this won't happen nothing to you, okay? You are eating genes, you are eating DNA. We are eating DNA. When we eat a steak, the steak is a muscle, and the muscle is made of muscle cells, and the muscle cells have DNA, okay? So we eat DNA, and there's nothing wrong with that. The problem is that if we create an animal or a plant which is resistant to everything, which is stronger, it can be a problem. It could be a invasive species, okay? The especies invasoras, okay? Those species can be harmful for ecosystems. Imagine that the corn, that plants spread their seeds, and if those seeds reach the environment, they will grow without control, okay? And maybe they are displacing the autochthonous plants, and they cause problems, as happens with the I don't remember how to say in English especies invasoras, and it's not invader species, it's another name, I forgot it. Pero bueno, eso, que al final se pueden convertir en especies invasoras. Ese es el problema que tienen los organismos modificados genéticamente, ¿vale? Las plantas, si llega una planta que resiste a todo, puede desplazar las plantas de nuestro ecosistema, ¿vale? Y lo mismo con un animal, y eso ha ocurrido, por ejemplo, con los bisones. Los bisones americanos hay bisones americanos y bisones europeos, ¿vale? El bisón que vive en Europa y en España es el bisón europeo. El bisón americano es el que se cría en las granjas para hacer piel para abrigos. Ha habido escapes de bisones americanos en Europa, y el bisón americano es mucho más agresivo y más fuerte que el europeo. Y al final, cuando hay dos especies que compiten por un mismo recurso, una desplaza a la otra. Y en Europa ahora mismo el bisón americano es una especie invasora y el bisón europeo está en peligro de extinción, porque el bisón americano le está desplazando, le deja sin recursos. Dos compiten, pues uno tiene que ganar y el otro tiene que perder. Y ese es el problema que hay con esto, con los organismos modificados genéticamente. Pero vamos, que no pasa nada por comerlo, que nos van a crecer florecitas en la cabeza por comerte un maíz modificado genéticamente, ¿vale? O por comerte un salmón modificado genéticamente, no te va a pasar nada. Vale, y esto es todo. El tema está acabado, y lo que quiero que hagáis es, de esto tomáis apuntes. No va a entrar en el examen, ¿vale? Os puedo preguntar para qué sirve una electrophoresis, o para qué sirve la PCR, y que sepáis, pues para hacer copias de ADN, o para identificar un fragmento de ADN, ¿vale? Eso os lo puedo preguntar, pero no hay que saberse todos los pasos que yo os he contado. Lo que sí quiero es que lo tengáis absolutamente todo en el cuaderno, y voy a mirar los cuadernos y lo voy a evaluar, ¿vale? Porque esto ya yo creo que es mucho intríngulis aprenderse todísimo esto. Pero con que lo tengáis en el cuaderno me vale. Así que eso, hasta aquí hemos llegado con el tema de genética molecular.